Thank you so much for having me as your last speaker of the decade. I know. Whew. My name is Yaamini. I'm a fourth-year Ph.D. student at the School of Aquatic and Fishery Sciences at the University of Washington. And today, I'm really excited to talk to you about some of the work that I do on oysters, ocean acidification, and omics. Now, I didn't choose my title just because it had a really fun alliteration, but I also chose it because I think that studying all three of these things at the same time is really powerful when we're considering how organisms are going to respond to future ocean conditions. And if you take anything away from my talk, I hope that's it. And before I actually proceed with my talk, I do want to acknowledge that we're on Coast Salish land, and particularly that of the Lummi and Nooksack tribes. All right, so let's get started. Oysters are an iconic part of Washington identity. Oysters are shellfish that live in salty water, so think oceans or areas where salt and freshwater mix, like the estuaries of Puget Sound. Oysters, within Washington, we grow 5 different species of oysters, or you may encounter them in some capacity. First is the Olympia oyster. This is the only native oyster species to the West Coast. But in the late 1800s, populations of the Olympia oysters were decimated, potentially due to overharvest or pollution from lumber mills. So we brought in Pacific oysters from Japan, so shellfish farmers could grow something and we could have something to eat. And again, these were introduced oysters from Japan, and although they were introduced, they're now an important part of the Washington shellfish industry. We brought in a couple other oyster species to cultivate: the European flat oyster, which is closely related to the Olympia oyster, as well as the Virginica and Kumamoto species. Virginica from the East Coast, Kumamoto from Japan, and these are closely related to the Pacific oyster. Today, I'm going to talk to you about my work with the Pacific oyster, which again was that introduced species we brought in the late 1800s, early 1900s from Japan that is now essentially part of the backbone of the Washington shellfish industry. Like many marine invertebrates, oysters have a really complex and interesting life cycle. So they start as adults. When they are good and ready, they do what's called spawning. So they send sperm and egg out into the water, and hopefully they meet and you get a fertilized egg. Fun fact, Pacific oysters are actually hermaphrodites, so they can start off as male, and over the course of their life, they will transition into becoming females. If that egg is hopefully fertilized, that egg will start to divide and develop into the planktonic free-swimming stage of oysters called larvae. Early larvae are what are called D-hinged because, well, kind of look like a D. And then as they grow and develop, they will start to become bigger, get some cilia, maybe be able to move around a little bit more, but they're still kind of at the whim of the tides. When larvae are ready to settle down, literally, they settle onto hard substrate like other oysters or harder surfaces, and we see these dime-sized versions of adult oysters, and these are called spat. And given 2 to 3 years, depending on water conditions, that spat will grow into the healthy, well-adjusted adult oyster we are all familiar with. So as they transition from life stage to life stage, or just grow and develop, there is a quote-unquote cookbook that they use and they refer to be able to essentially be an oyster. I want you to put a pin in that idea. We'll return to that cookbook later. Now, oysters have ecological, economic, and social value to us. Oysters are what we call ecosystem engineers. Out in the bays, they kind of clump together and create these reef-like structures, and these structures actually protect our shorelines from eroding during stormy weather. Oysters, just by the way they take up food from the water, actually can clean and filter our water as well. In counties, in a lot of Washington counties, shellfish farms and oyster farms are the #1 employer. And over 1/3 of profits from the Washington shellfish industry, which amounted to over $25 million in 2013, actually comes from oysters. And if we just broadly think about shellfish, shellfish have these social importances for several groups. In particular, tribal nations develop customs around harvesting and consuming shellfish. But the ecological, economic, and social importance of oysters is threatened by ocean acidification. So as we create energy, drive cars like I did to get to the seminar, or fly to parts unknown, we emit a gas called carbon dioxide, or CO2, into the atmosphere. Now, we need a little bit of carbon dioxide in the atmosphere to actually keep our planet habitable, but it's this excess carbon dioxide generated by human activity that poses the problem we're familiar with as climate change or global warming. But there's a second carbon dioxide problem. The oceans are a carbon sink and actually absorbed carbon dioxide from the environment, making it slightly more acidic. Scientists are able to measure the concentrations of carbon dioxide in the atmosphere and in the oceans, and one famous observatory is the Mauna Loa Observatory in Hawaii. They've been documenting this for several decades. So we can see from the late 1950s, the concentration of atmospheric carbon dioxide has significantly increased, and every single year you can see these headlines of maximum carbon dioxide in the atmosphere, 2018, 2019. We're just slowly building it up. That, again, carbon dioxide is absorbed by the oceans, and it's a little bit more noisy, but we can still see that upward trend. And then that carbon dioxide, when it's absorbed by the ocean, makes that ocean more acidic. So then we can look at ocean pH, which is a measure of how acidic the ocean is. And from the mid-late 1980s all the way to today, we've seen about a 0.1 unit decrease in the pH of the ocean. So this is just globally when we think about it. All right, 0.1 pH units. Doesn't really seem like a lot, right? But that small change can have a big impact. Because of the way the pH scale is designed, a 0.1 change in pH actually means that globally, oceans have become 25% more acidic since the late 1980s. What does that mean for organisms like oysters that actually live in this ocean? These are all the chemical reactions that occur when carbon dioxide is absorbed by the ocean. You don't need to know all of this, but there's two things I want you to take away or remember. One, we see this increase in hydrogen ions in the water. And this is what actually makes the ocean more acidic. It's that increase of hydrogen ions. Because we're making the oceans more acidic, it changes that environment to a point where oysters and other organisms that live in the ocean struggle to maintain homeostasis, meaning that they're struggling to maintain ideal body conditions so that they can survive. It's harder. It takes more energy for them to do this. The second thing that happens is that we get less carbonate ions and more bicarbonate ions. Carbonate ions are important shell building blocks for organisms like oysters. They build their own shells by taking carbonate out of the water and combining it with calcium. Right? So because of the chemical reactions that occur, we actually see way less carbonate because it's all being converted to bicarbonate. And oysters and other organisms like corals or other shellfish can't use bicarbonate. So it becomes more difficult for them to build their shells. Again, they have to expend more energy. And that difficulty maintaining homeostasis and the difficulty building shells is something that occurs at every part of that oyster life cycle. So let's say that fertilized egg is growing and developing as larvae. We are actually seeing developmental delays. So they're growing a lot slower, and they're not growing at the time scale that they should be growing. They can also grow poorly. So this is a really zoomed in microscope image of one of those D-hinged larvae, so one of the larval stages that looks like a D, and the shell doesn't look too great. It's got cracks in it. It doesn't look very strong. So even if they're growing, they're unable to grow well. As they transition from larvae to spat, they're actually unable to produce as much energy as they need to maintain their bodies because so much of it is getting diverted to just maintaining that homeostasis or attempting to build those shells. We see these significant reductions in calcification, so reductions in that shell building process even through adulthood. All right, so let's say you've made it all the way to the adult stage. You can still have a little trouble getting it on. Again, we're seeing these like varied fertilization successes. So you could put out sperm and egg into the water, but fertilization may not happen. And that's again because of energy. So they're spending so much energy attempting to just cope with these conditions that they're not able to divert enough energy into making really good gametes that can then be fertilized and then grow up. So at every point in the life cycle, we're seeing these negative impacts. But it can get even more complicated because what happens when these oysters are adults can affect how their offspring perform when water is really acidic. And this is what's called a carryover effect. So we have a lovely adult oyster. If this adult oyster is exposed to acidic, so low pH conditions, sometimes that can be beneficial for those offspring. And this is what's called a positive carryover effect. That parental experience helps the larvae because the larvae end up surviving better, they end up growing more, and they end up being bigger. So perhaps adult exposure could be helpful for their offspring, for their larvae. We can also see that parental exposure can be negative. So in certain species, when adults are exposed to acidic conditions, those larvae are much smaller, they're slower growing, and they have worse survival in those same acidic conditions. So we've seen positive carryover effects in oysters that live in Australia. We've seen negative carryover effects in clams and other shellfish that live on the East Coast, but no one has tried to figure out what happens to the species that live here. So that's the first part of my work. I decided to ask the question, how does ocean acidification affect multiple generations of Pacific oysters? So if mom and dad are living in these really acidic conditions, how does that affect the offspring? So to answer this question, I exposed 150 adult Pacific oysters to either low or ambient, meaning water that is just, oh my God, water that's just coming in, pH conditions. And these, so I was saying that the global oceans are kind of around pH 8.0, if you remember that graph I showed you earlier. Our pH, I just want you to first notice that these pH treatments are reflective of what's in Puget Sound, and that's already much lower than that global ocean average. So Puget Sound is actually acidifying a lot quicker than many other regions in the world. So knowing what happens is important. So after exposing these adults to either acidic or ambient, so normal conditions, I characterized their gonad tissue. So gonad is reproductive tissue, and I wanted to see if ocean acidification affected their ability to develop sperm or egg. So that's kind of like the direct effect of ocean acidification on oysters, on one generation of oysters. I then kept them in ambient conditions, so those non-stressful conditions for 12 weeks, kind of get them nice and healthy again. And then I did what's called strip spawning. So I took every oyster, shucked it open, and then collected either the sperm or egg and mixed them in concentrations that I could control. So I could see if larvae, 18 hours after those eggs were fertilized, I could see how they survived. So I counted the larvae 18 hours post-fertilization. And that was just to see if exposure as parents affected the larvae's ability to survive. So I'll start by just talking about that direct effect of ocean acidification on reproduction. All right. So I actually saw no differences between that low and ambient pH treatment when looking at reproduction, so looking at the development of those sperm and egg. And that's because all the oysters that I looked at were immature. They weren't developing. So when we characterize how mature oysters are, what we can do is we can take a sample of their gonad tissue, slice it super thin, stain it, and look at it under a microscope. And this is what's called histology. So these are examples of oysters that we looked at under the microscope. So every box is a slice of that oyster tissue. Since I have no clicker, let me see if I can do this. So this right here, this is an oyster that is actually not male or female. It is super immature. And then these are two oysters that are just starting to develop. They're like gamete.
Yaamini's not.
Oh.
Sorry.
Oh, the mouse isn't? Oh, that's fine. Okay. So A, that first panel that I can't reach because I don't have extendable arms, that is a completely immature oyster. B and C, those second two, those are ones that are just starting to develop sperm or egg. This bottom row right here, these are all female oysters that are slowly starting to develop their eggs. We know they're not developed because they're really small. So G, H, and I, again, these females are starting to develop their eggs a little bit more. J, we can kind of see larger eggs. So they're things that look like eyes in the slides. They have like a white center and then a big black dot. Those are mature eggs, but we're not seeing a lot of them. And then K and L are both males. They have gametes, those tiny, tiny purple dots. They had sperm from a previous reproductive cycle, but they have no new ones. So what this told us, we saw this for both of our treatments, and what this told us was that our oysters just weren't maturing. They weren't putting energy into reproductive development during our experiment. And that could just be because the water was too cold or something. And then we also saw no differences between males and females. So it wasn't like one sex was maturing more than the other. They were both just not maturing. So they were both a little immature, and that's totally fine. All that told us was that we couldn't really detect an effect. Now I'm going to show you those larval results. So this was after 7 weeks. We then kept them in ambient conditions for 12 weeks and then stripped spawned so we can get larvae and count them. And so now I'm going to show you those larval counts. And what we saw was this maternal effect on larval survival. So this y-axis right here is the proportion of larvae that were alive 18 hours after we fertilized those eggs with those sperm. And I'm going to show you data based off of female experience. So these are the proportion of larvae that were alive 18 hours after fertilization happened when the larvae's moms were in ambient pH conditions, so those non-stressful conditions. And here's the proportion of larvae that were alive after those females were in low pH conditions. So what we see here is that larvae, if mom was exposed to ocean acidification 3 months ago, then the babies don't survive as well. And that in itself is huge. One, because this was the first time that someone really looked at how ocean acidification affected multiple generations of Pacific oysters. But two, because there was that 12-week, that 3-month gap. And what that tells us is that what the parents experience, whether it be the day before they're spawned or 3 months before they're spawned, is still really important. There can still be an impact. And that's kind of scary, right? Because we don't know how long that lasts, but it's important to know. So I looked at this, and I was like, great, now what? Why did that happen? And this is where we can turn to omics. So what can omics, and I'll define that later, tell us about how Pacific oysters respond to ocean acidification? Right. So remember that cookbook I was talking about in the beginning? Oh no, slide does not look, okay, pretend the DNA stuff is not there. This is just a cookbook. That cookbook I told you about in the beginning, that's what we call DNA, right? Every living organism has DNA. That DNA has instructions for how to go about everything that a living organism needs to do. So if DNA is the cookbook, genes are the individual recipes. So there could be genes that tell oysters how to build shells properly. There could be genes that dictate how to create eggs and how to create sperm and when to spawn, right? They dictate everything. Now, we know that DNA is inherited by offspring from their parents. But we used to think that that cookbook, that DNA was passed down parent to offspring, generation to generation, unaltered. But we now know that that's not the case. So depending on what environments the parents live in, there could be little notes in the margins of that cookbook. Like, are you all familiar with Harry Potter? I don't know at what point Harry Potter will become a dated reference, but you know how Harry in the 6th book or movie finds the half-blooded prince's potion book, right? It's got like notes all over it. Think that. Like that's what's happening. There are notes in the margins. or entire sections of this cookbook could be highlighted or covered in whiteout, right? Those notes, those alterations don't change the recipes themselves, but they change how they're interpreted. And this is what's called epigenomics, right? So it's the changes that occur that change how genes or how recipes are interpreted, but they don't change those genes themselves. So obviously, like, adult oysters aren't making notes in little cookbooks to pass down to larvae. How does this actually manifest in DNA? So we've got our DNA again, and we've got our genes, so our cookbooks and our recipes, based on the environments the parents live in. So if they're living in these more acidic environments, that can add these little accessories or tags onto specific parts of DNA. And this is what's called DNA methylation, right? So it's just, again, that methylation doesn't change the gene itself, but it changes how it can be interpreted by the oyster. And what's even more interesting about DNA methylation is not that it happens, is not that it could change how oysters respond and how they're interpreting those genes, but the fact that those methylation patterns, where they are, what genes they're in, can potentially also be inherited by larvae when they inherit DNA from their parents. Right? So it's like getting a used cookbook instead of a new copy. If that DNA methylation is passed on, it could induce those carryover effects like the one I showed you earlier. Right? So that's why they're important. They can induce carryover effects, and they can change how oysters respond to their conditions. And this is important when we're considering ocean acidification or other changes to the environment. Right? Just really broadly, when we think about how organisms respond to their environment, they can either adapt, which is a process that takes many generations, or they can acclimatize, which is a process that maybe takes one generation. So let's say, for example, I took everyone in this room here and I plopped us all on Mount Baker, right? It's cold there right now. Over many generations, thousands of years, perhaps our body hair will grow slightly longer and longer and longer until we look like Sasquatches. And that's how we adapt to those cold conditions. But if we wanted to respond to those conditions right now, perhaps we put on a Patagonia fleece. Right? That's the difference between adaptation and acclimatization. And essentially, by studying DNA methylation, I'm trying to figure out if the oyster equivalent of putting on a jacket is helpful, right? Do they have jackets to put on? Are those jackets helpful for those conditions? That's what I'm studying when I'm studying DNA methylation. Do they have methylation, a tool that they could potentially use to respond to those really stressful environments? And is that response going to be helpful? Is it going to be a positive carryover effect? Or is that response going to be not helpful? Is it going to be a negative carryover effect? Is it going to be harmful? So that's what I am studying. Now, just to recap what we've talked about today, oysters are important, right? They give us ecological, economic, and social value, and we've got systems that depend on these oysters. Through my work, I've learned that ocean acidification can affect multiple generations of Pacific oysters. And I can use epigenomics as a tool to figure out why oysters are affected. And I really want to highlight the why. It's not enough to just know that oysters are affected by ocean acidification. It's not enough to know that what happens to the parents can affect the offspring in those conditions. Without knowing why those changes occur, we're not going to be able to fully comprehend or predict how these organisms are going to fare as oceans continue to acidify. Right? If we know why, we can apply that knowledge towards conserving their ecosystems or changing hatchery practices so that we're able to have oysters in the wild and at home on our tables. And we don't just have to study methylation in oysters, we can study it in other organisms as well. So I've mentioned that I do work with Pacific oysters, but I also work with Virginica. So with these two species, I'm looking at how oysters can acclimatize or respond to their conditions now using DNA methylation. In my lab, the Roberts Lab at the University of Washington, I've got lab mates that do other things. So I have a lab mate that studies methylation in the native oyster, the Olympia oyster, working with a nonprofit in the Puget Sound area. And I also have lab mates that work with the Jamestown Skolem tribe in Squim and work with researchers in Rhode Island to study how geoduck respond to ocean acidification using DNA methylation. It doesn't just have to be shellfish. We work with researchers in Chile to understand how Atlantic salmon that's grown there responds to parasites. And we can use DNA methylation to do that, to figure out on this molecular machinery level what's going on. And now we work with researchers in Rhode Island and Moorea to study if corals can use DNA methylation to be resilient to those changing conditions. Right? And again, it's all about learning why. If we care about these organisms and we care about the natural systems they're in and the human systems that depend on them, we need to know why they respond the way they do to be able to tell if they're going to not only survive, but thrive as climate change continues to endanger them. So I know this probably much shorter than you anticipated, but I figured there may be questions. So with that, I will stop. That oyster was shaped like a heart, so I thought it was cool. I want to thank you all again for having me and thank my various funding sources and collaborators. And now we just have a lot of time for questions to talk about oysters, ocean acidification, and omics. Thank you.